biologics and also the chemical, small compound, is a material, final product. But the cell therapy, final product, living thing, we can't kill the cell. Therefore, we have to have more information, more environment information, what kind of cultural information to make it good quality.
Hey, smart scientists. Welcome to another episode on the Smart Biotech Scientist podcast. Have you wondered why QbD-based cell manufacturing is necessary and how this could lead to more cost-efficient and more robust manufacturing processes? Well, then you're in for a treat today, because today I'm having a conversation with Shin Kawamata, who studied physics at the Kyoto University and medicine at the Kobe University in Japan and received a PhD degree then later in pathology. He moved on to develop a human leukemia mouse models at Systemics in Palo Alto, the USA, and Stanford University during his postdoctoral period. He became a chief scientist in the Foundation for Biomedical Research and Innovation and the director of the R&D Center for Cell Therapy. In April 2023, he became the CEO of Cyto-Facto in Kobe, Japan. Stay tuned for an exciting conversation. Welcome to the Smart Biotech Scientist podcast. I'm your host, David Brulman. Let's smarten up biotech. Welcome, Shin. It's great to have you on today.
Thank you very much for inviting me to this kind of talk today. So my name is Shin Kawamata. I'm a MD, hematologist and by background, and CEO of the Cyto-Facto at the CDMO for cell and gene therapy in Japan. So that is my background in here.
Absolutely. It's a pleasure to have you on today. And can you share something that most people don't know about you?
I am working in the field of this field. We used to work for the FBRI, the public institution, Foundation for the Biomedical Research Innovation in Kobe, which really supporting for the cell therapy in Japan as some kind of the public sector. And also we are manufacturing Kymriah of the Novartis as a commercial. We used to be last year, 220 batches were supplied to the hospital. So that is the thing, because the FBRI is a public institution, we have to leave. Because public institutions do not want to make money. But they also allow me to work in some kind of 50% of total income. But last year, so over this year, we exceeded the income. So therefore, it's the planned exit, planned spin-off from public institution, and named Cyto-Facto. Cyto means a cell. Facto is a manufacturing. That is a kind of the first, I think, in the history, first in a private company, CMO company from public sector in this. I hope it works. It is new business model because the government spend 15 years for the millions of dollars to support a facility for nothing. But I think it is something like outcome of the public investment. I hope we can work as more commercially to penetrate in the industry. That is our mission.
Yeah, that's exciting. And how did you get started in biotech before now becoming a CEO at Cyto-Facto?
Because I was a director of the cell therapy center, so automatically I have to be CEO. My boss was Dr. Honjo. He is Nobel Prize winner for PD-1 antibody. He is more like basic research. So they really asked me to leave. And I was a director of this site therapy center. Automatically I have to take care of the people. So 100 people was transferred to the Cyto-Facto as it is. And we succeed all project and facility activity as it is. So that's the reason why I become a CEO. It is not my intention. We have to do that.
So let's talk about the latest advances and cutting-edge research in the area, because you've been a long time in the industry and you know a lot about induced pluripotent stem cell-based cell therapy.
We used to work in some kind of regenerative medicine or using IPS and MSC for therapy, but I think CAR-T is the new area, new application for this one. And it's really linking with cancer therapy, immunotherapy, and gene editing, everything in it. And also the big pharma was also to join this kind of the field. Therefore, it is important to push forward this CAR-T or cell and gene therapy. Ex vivo, the cell therapy is the direction to go. That is my idea.
And what is your vision of the cell therapy industry?
Yes, the cell therapy industry is now growing, and I have to say very immature, and not like drug therapy, something like that. Therefore, we do need some participation, and not for the horizontal relationship. We do need some hospital, pharma, and CMO, the CDMO, should share data together. In order to share the data, we have to be digitized of the information we have. We have to be linked with their supply chain as well. So I think it's a new type of the modality to participating of this industry. It's not like biologic, not like small compound. So therefore, we have many room to activate, push forwarding of the cell therapy in this sense.
So there's still a lot of innovation coming ahead, right?
We are working very closely about the QbD-based approaches because, as you mentioned, that QbD was very famous in something 22 or something 20 years ago. But FDA was advocating about using of the QbD, this type of the approach. But it's not happened because you are working with the chemical and drug it. You don't need any quality by design. It was designed as it is. You don't need to connect it with manufacturing to the QC data. You can do that at the end of the day, just bind it. That is okay. So therefore, I think as long as you are like a material, you don't need any QbD. But if you are handling with living stuff, living thing, you need some kind of more monitoring, sensing to describe what you're doing. And also you have to combine the process to the QC data together. For example, so if you count your T cell and one day you don't get any T cell percentage, you have to be expand another one day. Therefore, the QC and the manufacturing should be linked together and also should link with inventory as well. So all of the information to get together. Otherwise, you can do any kind of the system for this one. So robot is one of the things we can do that, but robot is not clever. Robot do the mimicking what you're doing about it. So they have no intention to do that. Therefore, at the end of the day, the checking all of the quality, we call the verification at the end of the day for every shipment. That will really block industrializations. But we have to be guaranteed while you're manufacturing, okay, this is the system, and this is the protocol, this is a parameter, this is control value. As long as you have to get the result on the documentation, you can maybe verify the quality during manufacturing side. QbD approach should be needed as long as you are going to handling with living stuff. Its time has changed.
Yeah, absolutely. QbD is essential. And before moving into the nitty-gritty, the details of QbD, I'm curious, Shin, about the general manufacturing challenges, because we have great therapies, but the cost is still extremely high and we have huge challenges. So what are the biggest challenges and how can we overcome these challenges?
Big challenge come from many of the less information at many FTE. Labor cost something like 40 to 50% of the manufacturing site. We know about during the experience of the CMO businesses. But if you introduce some automation with QbD, with documentation, will really save the time and the money for this one. Because when you're going to making some CAR-T, we need a pile of paper, more than 1,000 of paper for one batches. And we have to hire 2 QA to verify, to check in about any mistake. But they still made a mistake. So because high cost, many papers, and still made a mistake, and Kappa, that really has another FTE needed. Therefore, by introducing automation and QbD digitalization really saves time and accuracy, and also visualize what you're doing. It is very important to figure out what kind of mistake, what kind of loss of the process, you can improve the process itself. And by linking with a kind of inventory supply chain, you will choose the most best scenario, best case of raw material, and should be kept back to the supply chain that really improve the quality of the raw material as well. And if you link the clinical data on the hospital, you can find out what's more important parameter to make a new drug, and in this case, that is the kind of thing we have many room to improve. But you have to be clever. You have to understand. You have to be introduced some more new identity, new idea as a QbD digitalization automation should be get together. It's not separate concept.
And something we focus on a lot on the Smart Biotech Scientist podcast is the commercialization, so the scale up. We need to deliver more therapies. And you alluded to it already a bit, but what are the key things we can apply to make a process scalable and especially more important to produce it in a robust way at large scale?
So it depends on allogenic and autogenic, a different story. But in any case of the allogenic, we may have more monitoring and verify the quality by sensing. That is the QbD approach. Really visualize the loss, visualize the point to improve. So that is the kind of thing we can introduce for the cost down of the price at the end of the day. And also it is very important to link the information by the crowd about facility information as well. So you can be optimize the time to change the facility or maintenance plan. Use the cost for the manufacturing cost as well. So you should combine all of the information together. But then the conventionally, we have the many software to linking together. It's like a one-to-one kind of the connection. You like a home-to-home telephone. Crowd type is something like the email. You send the mail in the crowd system. You can get some integrated information in it. You can just get out your attribute you needed. That kind of information should be integrated from the beginning. Therefore, we should have more new type of the software to integrate it, QbD type of the new eras. There are many room to do it. That's the thing that we are seeing very bright future in it. But this is lesson from working as a global pharma. When you're working as a global pharma, there are many sites around the world. So this is the US, this is Europe, this is the Japan. So they are compared the date every month, checking about the how can we standardize the globalization, how you stupid thing, what kind of mistake we made. So really pushing down the cost down. But really enforce me, and know that cost down, we do the innovation. Innovation also lead another cost down and the kind of the voice to deliver it, to linking with the pharma as a kind of the partner, not mass and slave. Therefore, we have to work with the global pharma. That's a very important thing.
As we move towards the QbD, the details of the QbD, and you briefly mentioned it already that the industry, especially the biologic industry, has still not fully adopted QbD, or is very slow at least, to be fair. Can you tell us, especially also to those listeners who might be critical about QbD, why is a QbD-based cell manufacturing actually necessary? And how does this ensure quality in the manufacturing process?
Biologics and also the chemical, small compound, is a material, final product. But the cell therapy, final product, living thing, we can't kill the cell. Therefore, we have to have more information, more environment information, what kind of cultural information to make it good quality. Therefore, more sensing technology, more indirect information may need it to constitute the quality of a thing. Therefore, we can't directly checking about the quality by the product. We have to be checking the quality by indirect by sensing. We have to need more like a QbD approach for manufacturing the living thing, like vegetable, fishes, and cell. Sensing technology was not catch up until recently. Therefore, QbD was just the word. But recently, and advancement of the sensing technology really catch up about QbD basis. Therefore, we have to apply new technology to just indirectly to visualize what you're doing as an image. We have to make as designed. That's a chemical compound you can make as design without any effort because it's very easy to control. For the living thing, you have to be carefully. You have to have an idea what is the QTPP, CQA, CPP. You have to set up the process parameter. You have to cut off the design space. Then you will verify sensing, okay, this is in the parameter, but in accordance with the QbD within the control design. Then we can verify in the time-wise and for kind of the long kind of culture period. Okay, we don't need the verification at the end of the day. This is the digitalized record for the verification of the thing. It's okay. You need some minimized testing for the sterile test, something like that, and lead it to shipping about this. It's a new type of modality may need it. That's the reason why QbD is needed for living thing.
And what are the specific quality requirements for mass culture of stem cells?
Depending on the iPS, MSC, and CAR-T is different slowly. In case of the iPSC, the mass culture iPSC has the QTPP, the quality target product profile should be something like a span, self-linear potential. At the same time, they have the ability to differentiate. But it is easy to verify it is also expanding as immature state, but it is very difficult to figure out do they have any differentiability of that. But the most important feature of iPSC has ability to differentiate because nobody use as it is. With differentiation is more important than the future of the iPSC. But I know that to address iPS and differentiation, we have to be care about the metabolism of the cell. A cell should be glycolytic, not OXPOS, not activate mitochondria. Mitochondrial activation means getting old, restarting differentiation spontaneously. Therefore, to minimize the activation of the mitochondria and accelerate the activation of the glycolytic pathway and tryptophan pathway. So like a Warburg effect, it's a very important effect for that. Therefore, in order to verify that, we have to be checking about the metabolism, and we buy the LC mass, and they are eating the sugar and secrete the lactose, and they eat the tryptophan, secrete the quinoline, not secreting too much adipin, and not activate mitochondria by checking the electron membrane. That's the kind of process parameter we have for the mass culturing in the process monitoring. That's the reason why for this one. For MSC, it's also the getting off your culture longer. So you have many culture for if you longer period of culture, but they have no pharmaceutical effect. But you have to be verified, this specified target population. So therefore, in the target population, they have maintained cell as self-renewal as a whole of the culture system. Therefore, we have measured the mitochondria as well. And if the cell was getting old, mitochondria fuse together, make a very long. They something a kind of sign of the aging. So the signs of mitochondria and the membrane electron and metabolism to use to maintain some redox system. That is thing we as a parameter to maintain as a stem cell. That is a thing. And the CAR-T is also very important to be more young because the CAR-T is a very important therapy within effective in the 2 months. But most important, it should be prevent recurrence at 6 months later. Therefore, CAR-T should be very young, survive longer in the patient. You should have some kind of the marker which is more younger and not exhausted marker like a PD-1. They have so very young and CCR4 and the CD28 and 27 and the CD26L, something like a marker, and metabolon and mitochondria was not activate all. So this kind of the parameter to manufacturing thing. So we do need sensing to qualify the quality.
Hey, smart biotech scientists. I hope you've gotten as much value out of our conversation with Shin Kawamata as I have. Shin Kawamata is the CEO of Cyto-Facto Inc. in Kobe, Japan. And in the second part of our conversation, we're going to talk about digitization. How digitization will make a difference in cell manufacturing and what the benefits are. And also we're going to look at networks and how we can leverage networks to drive down costs and make the whole manufacturing and development much more efficient. So stay tuned for part 2. Smart biotech scientists, I encourage you to take advantage of Shin Kawamata's offer for training. So do use the links in the show notes below. And if you want to unlock your full potential of your cell and gene therapy skills, I have great news for you because it's time to take your expertise to the next level. Professor Steve O and myself are crafting an exclusive training program that will ignite your knowledge and propel you towards excellence in cell and gene therapy. So if you're tuning in right at the release of this episode, can you spare just 5 minutes to share your training needs with us? Because your valuable insights hold the key to shaping the future of this field. And here's the exciting part. Be one of the first responders of our survey and receive a free 1-hour assessment of your organization's training requirements. It's a whopping $3,000 value. So if you want to join that, if you want to do that, reach out to me. You will find the information right below in the show notes. And if you're tuning in later, several months after the release of this episode, do also reach out to me, because we will already have developed this comprehensive training program in cell and gene therapy. So do reach out to me so I can give you all the information so that you can unlock the full potential of your cell and gene therapy skills. All right, smart scientists, that's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.